Okay, I'd like to thank everyone for being here. I'd like to thank the CNLM. I'd also like to thank my lab and also Kim Green. I hope you can understand me. I'm speaking British, if that's okay. So what I study in the lab, we all know that genetics plays a huge contribution to both early and late onset Alzheimer's disease. But we don't really know exactly what specific genetic variants do to specific proteins and then how they contribute to the disease pathology. So, my lab, we chose 2 proteins. These are called ABCA7 and ABI3, hence the name ABCs. And we looked at specific genetic variants that were associated with increased risk of developing Alzheimer's disease. And we then put these into some mice against their will, and we assessed what these variants did to Alzheimer's disease progression. And so interestingly, when we put in what was supposed to be a risk factor for getting Alzheimer's disease, it actually seemed to protect against Alzheimer's disease. So this really reiterates the point of, even though we can sort of associate specific genes to different risk for developing AD, doesn't necessarily mean that that's what happens in mice and also humans. And then secondly, in the second protein, ABI3, we actually did see a significant difference in immune cell function. So the future work that we really want to do is, our animal models for AD are rubbish. They are terrible. And so what we're trying to do is we're trying to generate better models, specifically mouse models, to recapitulate human Alzheimer's disease. And so one way we're doing that, we're going to introduce some of these variants into these new mouse models, and we'll see how these affect pathology. And if you're interested in our development of these models, you can look at modelad.org. It's a US-wide consortium. And yeah, that's about it. Cheers.